And so the story unfolds further, and it's my very great pleasure to introduce to you Professor Guillaume Lessene. Guillaume, I don't know how you happened to come to WEHI, so maybe you can tell us. He came to us as a wonderful chemist to join Peter Coleman and his team from France. So he's got a great accent. He missed out on that wonderful Hawaii meeting, but he impressed us all with his enthusiasm because it was the deep, dark cold of Melbourne winter, and he got up at 3 in the morning so that he could join in. So we knew he was keen. He's played a huge role. He's going to speak on behalf, I think, of our medicinal chemists and structural biologists who picked up the baton and took it from the mouse all the way to being a possible drug. Guillaume, welcome.
Thank you very much, and it's a very great honor to be able to present some slides and talk about the depth aspect of the collaboration. So just to talk about synergy and a series of firsts that happened throughout this work, and also luck, because the reason I'm here is because of my wife. And for 2 reasons, too, because I came to Melbourne, but she also sent me the ad for the position here at WEHI, and I applied, and the rest is history, at least for me. So that was fantastic. So I'll start with you, Susan, because this is, I think, the key part of that part of the journey was the decision with you and the board to decide to go into translation and to bring, to leverage on the knowledge and expertise of new people that were actually new at the Institute. So structural biology with Peter Coleman, high throughput screening with Ian Street, and the medicinal chemistry, which I joined on December 2001, and just put some personal pictures of Jonathan Bell and Keith Watson, who headed the medicinal chemistry lab when I joined. So obviously it's been a journey for us medicinal chemists, starting completely new into this institute and getting to interact with people here. And obviously as a chemist, I had very little knowledge about biology in general. I was interested, but had very little knowledge and very little in apoptosis. And really through the interactions with people like with David, Andreas, Andrew Roberts, really got to really know the field and really be passionate to develop compounds that target the pathway. So just to give you just a very quick overview of what all these people and all these groups contribute to the medicinal chemistry pathway or journey, but also in general for biology. So high throughput screening at WEHI started with Ian Street. Really, the idea behind high throughput screening is really what I've written here, is how to find a needle in a haystack. So where can I find this starting point to start medicinal chemistry that will lead to a potential drug? So this is really done through, and we were talking about earlier on about technologies such as screening, but also the talent and expertise of people that can design the right experiment to really sift through this quite large library. And again, to talk about first, this was the first high throughput screening lab facility in an academic environment in Australia, and probably one of the first in the world at the time. So really pioneering the idea of trying to do this like a pharmaceutical company in an academic environment. And as you've seen, and that was also really important and for the future, really all this work was based on incredibly talented biologists that we were all fortunate to work with, and which really are the reason why this project really progressed so well. Structural biology is really this ability to look at the atomic level at protein structures, but importantly for us medicinal chemists to be able to see how a compound are binding to these targets, and therefore not only see how they bind, but also think, how can I change that small molecule to make it more potent, to make it more, to interact better with these proteins, and therefore increase its potency, etc. And obviously this was enabled by access to the S0207 synchrotron here directly on our doorsteps in Melbourne. And finally, medicinal chemistry. So this is sort of integrating, certainly also, although a lot of biology was known, just now looking at the biology, but with compounds involved. So How do you go from that needle that was found through the screening all the way to something that's we would call the lead, something that looks a little bit like a drug, but that still needs some work. And that's really integrating a lot of data around these small molecules, which obviously is biological activity, is the compound working in the models that we're interested in. But we have to also always keep in mind the fact that we want to develop a drug. And biological activity is only one side of the small molecule. It's also the properties that are important. Therefore, is the compound reaching the target in the body. And this circle here happens many, many, many times until we can say, yes, we've got a compound that now we can test in a proper model. So this is what medicinal chemists were doing in the lab here in Bundoora initially. So here is where all this was set up, and we were really fortunate to have fantastic labs to work in, which, as you see, that's a very early picture of the facility, which became this really fantastic facility, and we now have laboratories here directly in the Abbvie campus. And this was also hosting the high throughput screening. So this really enabled this ability to translate the biology from the bench research all the way to small molecules. So what are we trying to do? As I said, we're trying to bring biology all the way to small molecules to drug leads. And we interact strongly with structural biology, and this is the process of medicinal chemistry. But obviously what this whole process brings into the mix is that all these components interact with each other, and now small molecules can help understand biology, structural biology can help understand biology as well. And this really, at the end, and like the proof of it is now, where all these components that were very new in 2000s are now completely integrated into WEHI as part of the basic research. So what we were really fortunate to start with, and obviously that was my first project, and that was another fantastic stroke of luck to be able to work on this project. Starting from the BCL2 family biology, as was shown by Dave, Andreas, and David, and many other colleagues, what we worked on, and I have to emphasize that this part is actually before the collaboration with Genentech and Abbvie, and we worked around developing inhibitors of this congener of BCL2. So BCL2 has many siblings in the family, and BCL-XL is another important one in cancer. And we're really interested in developing compounds targeting that particular pro-survival protein. And I'm just showing here some key elements. And I think one personal reflection on this, and that was before the collaboration again with Genentech and Abbvie, is that already around the table you had the world-leading biologists in the biology of BCL2. You have structural biologists. I've mentioned here clinicians such as Andrew Roberts, because already at the time, and we were still very far from the clinic, but your insight into what patients would actually like, what the type of drug that would be desired was an incredible insight even earlier on in this journey. And which probably has become quite important in other projects now. Medicinal chemistry, I've mentioned all the people that were involved throughout the project here and the screening. And all this around this table thinking, wow, this is a team that a pharmaceutical company or a biotech company would like to interact with, because there's really the top scientists, at least in the biology, structural biology, working around. And we developed internally some very important compounds, and we're part of the journey that enabled us to interact with Genentech and Abbvie. I just want to highlight one key moment, because we have to realize that you work for a while with compounds, you make new compounds, they are more active, and then suddenly you make one that you're able to crystallize. And Peter Sabatar, who had joined Peter Coleman's lab, saw this first crystal structure, and this really opens an amazing amount of possibilities of understanding or clarification about how the compound binds and what you can do with it. And really, I think we had champagne on that day. I don't have any picture, unfortunately, but this was, I think now we've come to expect that we will get a crystal structure of a compound, but these are very difficult targets. This was first in many reasons, and this really was fantastic. And we were able to publish that many, many years after that. So then came the collaboration. So that really, again, synergy, collaboration, but on an extended level, creating many interactions, friendships, so with John Flagger, Andy Sowers, Wayne Fair Brothers, really an amazing interaction, scientifically, humanly, and obviously we worked, and this really was an enormous team, and I remember one quote from Andy Petros, who was one of the early scientists at Abbvie, working in this field and participated in the development of ABT737, which was the real first inhibitor of the family, saying, wow, these three institutions together, this is a formidable team. It's just the best team in the world. And indeed, so Abbvie had obviously had an enormous experience both in the basic science of BCL2 family protein and had gone into that field very early on. And Genentech oncology research really wanting to enter this field as well. So really amazing team. And again, very fortunate to be part of this, the next step of that journey. And we worked on these inhibitors of BCL2, BCL-XL, and MCL1, which are all part of this pro-survival family. Another interesting thing, so during this collaboration, really, people were working on the project all the time, and we would wake up in the morning, get on emails or phone calls from Abbvie saying, on Genentech, oh, this is what we've done, and then we would take on the work and then send an email at night to our colleagues in America, and the cycle would go like this. So it was just an amazing experience as a young medicinal chemist to be part of this interaction. So I just want to highlight a few points, and this is the first point is true before and after, before and during the collaboration with our Genentech and Abbvie. Really having a tight team, and David alluded to that, having a tight team focused and committed to do this translational journey is essential, and obviously the biology itself is absolutely crucial, and we are really fortunate again to have world-class biology here at WEHI. This also demonstrated that academic teams can effectively work with pharma companies, biotech companies, and really drive academic drug discovery. I think this very vague point was actually illustrated by David just before. Infrastructure, it looks like a real simple thing to register compound in one place and another. It took a year and a half to have that infrastructure put in place. It was an enormous amount of work to actually have all the databases communicate with each other. Again, we talked about scientific rigor earlier on. This was a constant, not only in the basic science, but also in the development of those small molecules. We learned a lot interacting with very different partners. Obviously, Abbvie and Genentech are very different companies, biotech companies from the South San Francisco region, and Abbvie from Chicago. One is a real pharma company based on a century of medicinal chemistry, one based on excellence in biology, really two different atmosphere and drivers that we learn from, and we definitely have taken a lot of learnings from what we want to do and how to interact further with companies. And also, as I alluded to, be prepared to wake up very early or late, depending on who you're collaborating with, to be able to interact. Obviously, Australia is in the middle between all this, so we have to wake up early. One key moment, and we've gone through a little bit. The photos are not that good, but this is one thing that I remember. We came from Portugal, we landed on the 4th of November, put our bags in our hotel room, got the train a little bit reluctantly because that was already a pretty much 20 hours day from Portugal with the jet lag, but this is the ticket that I still have, and really this special event that probably feels a little bit like another world now on that day. And so the adventure continues for us. Obviously, this was a successful example, and we want to do that again. We want to do that with more projects, with our key therapeutic targets area here, more screening, more molecules, and more collaboration with companies, and hopefully more drug leads, and hopefully, again, more drugs in the market. Thank you very much.